have sickle cell, your blood cells are not able to carry oxygen as well. So it's hard to oxygenate your tissues because your red blood cells can't bind to that oxygen very well. And so this sickle cell is actually caused by one mutation in one nucleotide in the DNA. And that's what this figure is trying to illustrate. So in this case, here's our normal DNA strand. All right, this is the normal. There's no disease here. This is how it's supposed to be. So we have our DNA sequence right here. And that DNA is then going to be transcribed into RNA. And that RNA is going to be translated so that you're going to get a series of a proline, a glutamate, and a glutamate. And that's not the entire protein. That's just, we're just looking at this one little section of protein. But in sickle cell, what happens is what was a T right here gets mutated or changed into an A. So if you change that to an A, that means instead of an A being in the RNA, now you're going to have a U. And so by changing that DNA, it's going to affect the RNA. And then by changing that codon, when the ribosomes come in and want to put in an amino acid, it's going to do it based on the codon. And so it's going to put in a valine instead of another glutamate. And so just by changing one amino acid in this sequence that makes up hemoglobin, which is the molecule responsible for carrying oxygen to all your tissues, when you have just one amino acid changed, that can give you sickle cell. So all we did was change one little nucleotide and you end up with a disease. All right, so not all diseases are that simple. Other diseases are more complicated. You have multiple mutations in multiple genes. But this is an example of a simple disease in which you get the disease just because you had one nucleotide that was incorrect. And that affected your RNA, and then the incorrect RNA affected the protein so that you don't have the proper amino acids in the proper order. So DNA...
How does that even happen?
Mutations can happen various different ways. They can be spontaneous, they can just like randomly occur. They can also occur during DNA replication, which we haven't talked about yet, but when a DNA needs to make a copy of itself, we call that DNA replication. And it's based on the idea of complementarity. So again, if I have a sequence that's, let's just make it simple, a bunch of A's, and I want to replicate my DNA, I'm going to do it based on complementarity, right? Well, sometimes that enzyme that's responsible for making the copy of DNA, it'll actually put in the wrong one. And so it can be a random mistake. It can happen during during DNA replication. It can also be induced through mutagens or carcinogens, so things that cause mutations like UV light, smoking, drinking, radiation, sitting in a tanning bed, all day long. All those things can induce mutations. And so there's multiple different types of mutations, which is what we're going to focus on. And I want to get through this so we can watch that video. Yeah?
So how does it know it's a mistake? Does all hemoglobin have the same?
Yes, because all genes, all DNA, so my DNA for the hemoglobin gene is going to look nearly identical to your DNA sequence for hemoglobin. That's why when we sequence the genome, we have the correct sequence of every single gene, which means we know what all of the amino acid sequences are going to be that correspond to that. Yeah, so we know what the correct one's supposed to be because we've sequenced the genome. And so we know what all the correct versions of the genes are. And now there's databases of all these mutations. So all you literally got to do is go online and look up and see what it's supposed to be and then compare your sequence to that to see if there's any mistakes. And so there's different types of mutations depending on how that amino acid is affected. In a silent mutation, you're going to still have a mutation and it's still going to change your RNA, but it's going to change it to an amino acid that codes for the original amino acid. So you're still mutating it, But remember we said multiple amino acids can code for the same, multiple codons can code for the same amino acid. And so in a silent mutation, all you're doing, you're still mutating the DNA and the RNA is still changing, but you're ending up with the same amino acid as you would before. Now, if you have a mutation that's going to change the amino acid sequence, so just like in the example of sickle cell, we call that a missense mutation. And then there's also a nonsense mutation. This type of mutation is going to generate an early stop codon. And so if you change the DNA and that changes the RNA into one of those stop codons, then you're going to end up with a protein that's cut off before it was finished being made. And then there's also frame shift mutations, and when we're talking about the frame, we're talking about how those ribosomes read those codons, those 3 sets of nucleotides. And so, briefly, here you can see your original DNA sequence. All right, so this is what it's supposed to be. Now, in a frame shift, what's going to happen, you're either going to insert an extra nucleotide or you're going to remove a nucleotide. And so by deleting one of these nucleotides, in this case we've inserted an extra one in between this C and this G. So when we insert that, we change the reading frame. So this reading frame is that right there. There's the second codon. But if we insert an extra nucleotide, then that's going to change this codon. And that's going to change which amino acid gets brought in. So we call this a frame shift because we're shifting the reading frame. When we're talking about the reading frame, we're talking about codon followed by another codon. So here the codons are GAC, GAC. But just by inserting that extra nucleotide, now our codons are GAC, TGA. And so everything downstream, everything from that point on is going to have the wrong amino acid because we've shifted everything to the right by 1. Just like if we inserted 2 extra nucleotides, then we're going to shift the reading frame even more by 2 nucleotides. And so again, that's going to affect all those amino acids. Now if you have an insertion, and by the way, this is just looking at insertion, but if you delete these nucleotides, it's going to be the same thing. It's going to change the reading frame, so it's going to change your amino acid sequence. But if you get an insertion or a deletion in a multiple of 3, so in this case we've inserted TTT, it's not going to change the reading frame because this codon is still going to be correct, and this one, you're just inserting an extra amino acid. And so these frame shift mutations are going to be caused by either an insertion or by a deletion. And that's going to change the way that the ribosomes reads those codons and is going to dictate which amino acids are going to be brought in during translation. Michelle?
Are they always inserted at the same spot?
No, I mean they can just be inserted randomly. Just randomly, yeah. And so I don't know, let's see. Okay, so this might help y'all. It confuses me. It's kind of like the whole brownie analogy in the cookbook. So I don't know if you're going to like it or hate it, but I'm going to give it to you nonetheless. So what this is supposed to say, they're using the 3 codons to make a sentence. So each one of those, you know, the THE obviously spells the, but that's supposed to represent one amino acid. This is supposed to be another amino acid. And so when you read it correctly, it should say the one big fly had one red eye. And so in a missense mutation, again, you're going to change that nucleotide so that you end up with a different amino acid. So now it's not going to read the same as it should. In a nonsense, yeah, you're going to get that early stop codon. So you're going to be missing the rest of the protein. And then in a frame shift, so the one you inserted this extra Q right here, so it's going to change the way that that codon is read. And so everything else is going to be messed up behind it. And then if you delete 3 letters or add in 3 letters, you're just taking out, in this case, we're taking out fly. And so it reads the one big had one red eye. So you still got most of the amino acid, but you're missing that one. And then you can also have this insertion, which is the same thing. It just adds a new amino acid to it. Yeah?
Will I need to know every single one of these mutations on the test?
Of course you are.
This makes sense.
Yes, good.
All 8 or 9 of them?
Okay. All right, so if you want to stick around and watch the translation video, that's fine. If not, I will see y'all on Wednesday. Homework is due next Monday.